So today we are going to review a little bit for your final exam next week, and we cover chapter 21 and 22. And I would like to start with you on some low-stake assessment question, because we talked about the topics, and I hope that you remind some of the stuff. So let's use our self-assessment cards. You know how they work. So quickly remind you about it. You will take a coin, and each question on the self-assessment card relates to a question on my PowerPoint presentation. So, for example, question 1 has 4 different response options, A, B, C, and D, which is also an option on your self-assessment card. It's A, B, C, and D. So please take one of these coins, and you can choose what is the answer. Is it A, B, C, or D? Then you scratch off the field. And if it's the correct answer, you will see a star. You can assess yourself with 5 points and write it on the right side. If it's wrong, it's not a problem. Just give a second try and maybe you get 3 points. And you can keep continuing on until you find the correct answer, and then we discuss about it. Does everybody has a coin? Yes? Okay. Thanks. So question number 1 is about a genetic linkage map is based on what? A, it's a physical information from sequencing data. It's the actual number of nucleotides between loci. It's recombination frequencies to get the relative distances. Or it's B and C together. So think about it. What do you think is the correct answer? Correct answer is C. A genetic linkage map is not based on physical information. Physical maps are different. Physical maps are based on your sequencing data. You have the actual number of loci, the actual numbers of SNPs. Our genetic map is based on calculating a recombination frequency, like pairwise recombination between 2 markers, and then you can calculate the genetic distance and you set up your map. Question number 2. If you would like to create a mapping population, what do you need to do? Do you need to select parents which have no variation for your trait of interest? You cross and you develop your desired population. B, you can use a sorghum diversity panel, for example. C, you are selecting parents having variation, you make your crosses, you develop your population. Or D, none of the above. And don't forget to give yourself points. If it's correct the first time, you get 5 points. If it's correct the second time, you can still assess yourself with 3 points. The correct answer is C again. It's very, very important that you select parents that have variation for your trait of interest. High yield and low yielding, for example. You need genetic variation. You cross and then you develop your population of interest, maybe an F2 population, a recombinant inbred line population, or a near isogenic line population. Now let's talk about it. Why are maybe recombinant inbred line populations more important than the F2s? Why are they more desired? And we talked about it in class, and maybe you can remember it. Does recombinant inbred line populations are desired because they have fewer recombination events? Is an F2 desired because you have smaller linkage blocks, you have more recombination? Or option C, do you have a recombinant inbred line population more desired because you have smaller linkage blocks, you have more recombination events, and the progenies can be replicated? And the option D is that your F2 population is easier to generate and the results are reproducible. Try to remember what we talked about it. What do you think is the correct answer? C. So if you remember when we talked about it, if you create an F2 population, You are crossing two parents, you have that F1, yourself, you have one, you have your F2. So you have not much recombination going on in comparison to a recombinant inbred line population because you're selfing your F2 lines over several generations. So in your recombinant inbred line population, you have way smaller linkage blocks. Due to the selfing, due to the recombination, you are breaking down these huge blocks into lots of small ones. And this is desired, because now you can narrow down your causal gene being linked to your marker you may be found in your QTL mapping study. And the advantage is that you can replicate the progenies, because now you have selfed inbred lines which do not segregate anymore. And question number 4, I was wondering if you remember, how would you create a genetic map and use it in your QTL analysis? We just turned you the homework back today, and in your homework you did a QTL mapping study. How did you create a genetic map, if you remember the R code? Answer A, B, C, or D. Is it important to calculate recombination frequencies? Is it important to have polymorphic, non-polymorphic markers? Do you need sequencing information? Do you need a mapping population, or could you use a sorghum diversity panel? The correct answer is B. If you want to create a genetic map, first of all, you create a mapping population and you need to screen it. It's very important that you have polymorphic markers. If you cannot see a difference between your markers and your trait of interest and being linked to the causal gene, you have difficulties to create a genetic map. We used R in class, and in your homework assignment you did it as well. And you calculated first recombination frequencies. You then calculated the genetic distance. You somehow ordered your markers based on linkage groups, so you remember that. And then you built a genetic map. So your map is basically a distance measure, and it's a probability of a recombination in the interval between two loci. So you calculate pairwise recombination. And then you, based on the recombination frequency, you set up your map. So your markers who are closer together have a less probability of having recombination happening between of them than markers which are further apart on your genetic map. Does that make sense? Now we're going a little bit more in detail. What is a mapping function? And we talked about it in class. And we were wondering, is a mapping function a model that tries to relate the genetic map distance to a physical map distance? Is the relationship between a genetic map and phenotypical data? C, it's a model to explain how a genetic map relates to phenotypical data? Or D, it's a mathematical relationship between recombination fraction and genetic map distance. What do you think?
Say it out loud.
If you want, sure.
I think the answer is D. Just because when you don't use a mapping function, then you're not taking into account for interference potentially.
Correct. So the answer is correct. So let's talk about this in a few seconds, or in a few minutes. Before we talk about this more in detail, there are typical mapping functions which are used when you do QTL mapping. And this is Haldane, Morgan, and Kosambi. And before we go more in detail also what Seth just mentioned, I would like to know from you, what is the difference between those mapping functions? Does Kosambi assumes crossover interference and Morgan does not? Or does Morgan assume crossover interference while Kosambi does not? Is Haldane maybe a mapping function which generates more accurate maps? Or D, does Morgan assumes linear relationships between map distance and recombination fractions, resulting in the most accurate genetic map? This is now a pretty difficult question. It's not a low-stake assessment question, but we will talk about it. The correct answer is A. Kosambi assumes crossover interference and Morgan does not. And now I want to talk with you about it. If you have a look on this slide, on the left side, you see a graph which basically has on the x-axis the map distance in centimorgan, on the y-axis the recombination fraction. And if we go back in history, originally Morgan assumed there is a linear relationship between the map distance and the recombination frequency you calculated. So he assumed you have two markers, you calculate the recombination frequency, and that linearly relates to your genetic map distance. And then there were two researchers who said, I disagree. This is not true because we have crossover events happening. And this is Haldane and Kosambi. And Haldane was the first one who said, we need to use interference. And interference means that this is the effect of a crossover in one region on the likelihood of a crossover in another region. When he calculated and used his mathematical relationship, you see it's not a linear relation anymore. What you can observe in the short distance down here, all these three models have a linear relationship between map distance and recombination fraction. But the further apart we are, it's not a linear relationship anymore. And this is where Haldane comes in with his mathematical model on crossover assumptions, but he is not using interference. So Haldane assumes that crossovers randomly happen along the chromosome. And Kosambi said, oh, one moment, this is not completely true. I like to use interference related to crossover, that it's not always randomly, and he uses a different mathematical relationship. But we don't need to go more in detail. I just want you to see these are basically the differences. And maybe you remember the genetics lab. Mm-hmm.
Both functions, the Kosambi and Haldane, those converge at 0.5, right? For recombination?
The maximum recombination frequency is 0.5.
Okay, so they converge?
It cannot go, yes, cannot go higher. It's 50% always. So if you remember the genetics lab, and I put that slide in, and you remember meiosis. For example, why do we need to account for crossover? At the very beginning, you cross two parents, you have meiosis happening, and at the very first stage in meiosis, you can have crossover. If you evaluate your 4 parental cells, you can have 2 parents, two no crossover phenotypes, which are your parental types, and you can have 2 recombinants. Now they differ in their phenotype. They are not the parental types. So if you calculate if you calculate recombination frequency, now you count the recombinants, which is 2, over the total of 4, and you have 50% recombination frequency. But if you take double crossover into account, what you see here, if you focus only on your phenotype, red and tall, white and short, and you have a double crossover in between, you would not be able to see it, because all your 4 cells down here would appear as parental types. And this is the point where Kosambi and Haldane comes in, because they say we need to account for this. Otherwise, this is why the genetic map distance is not linear to the recombination fraction. And the last question, question number 7. Which methods of QTL detection can be used? What do you remember from class? What did you do in your homework assignment? Single marker analysis, a double marker analysis, interval mapping, composite interval mapping. What did you do? A, B, C, or none of the above? The correct one is A. You did a single marker analysis. We also did interval mapping and we did composite interval mapping. So these are now the most important topics you should know from the chapter and about mapping, mapping functions, QTL mapping. I would like to talk to you now about a genome-wide association study we did in class and in your homework assignment, and I would like to group you up. Because I want to talk with you about how can we find marker trait associations and how do we practically do it. So I want you to group. For group number 1, it's the question about what are typical populations and input data for GWAS. And I would be happy if Melinda and Kyle and Chance could be a group. For group number 2, do we need to account for certain information? Here I would be happy if Robert and Peyton and Carson could be group number 2. Group number 3, a general linear model needs which type of data? Who is left? I take Jada and Fatima. And the last one, what is the key concept of GWAS, is Seth and Diana. So I would like you to take a few minutes, and I would be happy if group number 1 could work here at the board. You can take the big white papers. Here I have pens for you. You can take notes to answer your questions, and then we will discuss about it. Group number 2, 3, and 4 can go over here.
That's your hand, right? Thanks very much.
You don't like this? So the main thing to do on it is that it's an unstructured population. Somebody has lots of natural variation at the university. It's not like you have a fucking cause where you're saying also.
You are maybe one. Oh, yeah.
Well, at least for our trade diversity.
What did you account for?
Population structure.
You mean in terms of like phenotype, genotype? What do you mean?
No, they should be tied together.
You just have to have the same, 1, 2, and 3 are here, 1, 2, and 3.
Yeah, that's what I meant. Two kinship.
Do you feel comfortable on talking about already?
Two couple data analysis calculations.
What are your findings? let's talk a bit and start with group number 1. The question for group number 1 was, which populations could I use for a genome-wide association study, and what as well?
So we said for populations, diversity panels, and not a mapping population.
So the key is that it's an unstructured population that relies on existing variation. It's not something where you're planning it out and making crops or something like that.
Exactly, unstructured populations like he mentioned, and you are using like a diversity panel because you have way smaller linkage blocks. You can assume many, many generations of recombination because you are considering ancestry and historical recombination events as well. Which type of input files do you use for your genome-wide association study?
You need your genotype and your phenotype information. You mean more specifically?
No. So he said we need a genotypic file. Your genotypic file is usually based on what? Which type of sequencing do you use?
SNPs.
Yes, and it's a GBS file, correct?
Right.
Then it's correct, you need a phenotype file. So you score your diversity panel, and then you use these as input data. Correct. Group number 2. And what your question was about, do we need to account for certain things to avoid false positives?
Yeah, we said yes. And the things you need to account for would be population structure, the kinship, and have diversity within the trait of interest.
That's correct. So basically, what is population structure and kinship? What are you calculating there?
Well, kinship is a relatedness between individuals and generally expressed in a matrix, whereas population structure is expressed in a principal component analysis.
Very good, that's correct. These are both relatedness calculations, and in a population structure, you basically structure your entire diversity panel in smaller, more related groups, and your kinship is a pairwise calculation between individuals to get relatedness data, and here you use a correlation matrix. Correct, and kinship goes as a covariate into your GWAS model. And now we know what we need to account for. So if we are building now our model for a genome-wide association study, what did you find out in group number 3?
So the general linear model combines the phenotypic data, the genotypic data, as well as the population structure, which is the principal component analysis, the PCA. Whereas the mixed linear model takes in kinship into account to add that relatedness for an association.
That's correct. Yes. And kinship goes in as a random effect. And group number 4, what is the most important or the key concept for GWAS?
So we determined that it's linkage disequilibrium, and this is just basically, SNPs are in linkage disequilibrium when they are inherited together, and that's called like a haplotypes. And what you want is that your gene of interest is in linkage disequilibrium with the SNPs, with some SNPs that you can detect by GWAS.
So please come here so that we have a look together on the last slide. The answer was correct. GWAS is based heavily on linkage disequilibrium. And what you see here on the slide, basically your genotype SNP. Down here, this is your GBS marker in your genotype file. And you are associating your phenotype to your GBS markers. But it's only an indirect association because this genotype SNP is assumed to be in linkage disequilibrium with your causal gene. Linkage disequilibrium means it's a non-random association of alleles. So you assume that your marker, it could be the causal gene, if you are lucky the marker is directly in the gene, or it's in association to your causal unobserved locus you want to detect. This is why we say the indirect association. It's a significant association which is not necessarily causal. It's very important to keep in mind. And also you remember in class I told you LD is not equal to linkage. LD means it's a non-random association of SNPs, of alleles, sorry. So you can have your marker and your causal loci next to each other on the same chromosome, but it can also happen due to history or ancestry that SNPs or alleles on different chromosomes are in LD because they appear together due to structure. And now, to avoid that you have false positives, you need to account for your population structure, because if you have too much relatedness, you might get a positive result because your population structure gives you a positive result, but it's not because you are associated to your trait of interest. Do you have questions on what we just covered? Or do you think you feel comfortable on understanding the key concepts about QTL mapping and GWAS?
What are some of the most common reasons that people have mistakes or trouble when doing a QTL mapping situation?
You mean when you are doing QTL
When doing
mapping?
QTL mapping, what are some common errors that people make that are good to look out for?
Errors. I would assume you need to do proper genotyping. So if you don't have a polymorphic marker and you cannot proper score your recombinant inbred line population for this is marker A, this is marker B, you might not be able to associate your marker to your phenotype, because you are basically in this single marker analysis, you are regressing your marker to your phenotype. Or if you do improper phenotyping in the field. I think all what matters is how you set up your data set. Are you really, when you analyze for your markers, are you really able to score your bands on the gel? Can I really see a difference? And usually you take your parent 1, your parent 2, then you maybe look for a certain marker. You know parent A has maybe a smaller band signal, parent B has a larger DNA band. This is how you can score for A and B. And it needs to be very easily scorable across the entire population. Does it answer your question? Or did you mean like, what were the mistakes students make in the homework assignment?
I know, you answered my question.
On question 7, you asked about single marker analysis, and I think you just said it. So single marker analysis is when you're just relating that marker to the phenotype.
So in single marker, you're basically regressing one marker and your phenotype. When you do an interval mapping, interval always means it's a region on the chromosome. Interval means you are involving 2 markers. You could also do a single interval mapping, that means a single interval. It means 2 markers, right? Flanking an interval. And there you are also regressing that single interval to your phenotype. The time is over. Good.